So good morning, everybody. I hope that also those that are connected online can hear me. Can someone tell us from the online people that it's correct? Hopefully, yes. Welcome to the second day of Physiology and Medicine that we organize here at the Institute of Physiology for undergraduate students. This event is organized with the aim to introduce you available positions for your bachelor, diploma, or PhD studies. My name is Olga Zimmermanova. I am a vice head of the Laboratory of Membrane Transport, and I am also responsible for all outreach activities of the Institute. And first of all, I would like to welcome here the director of the Institute of Physiology, Dr. Jan Kopecký, who will tell you a short introduction about the Institute, what we study here, and what we propose.
Thank you very much, Olga. Thank you, everybody here. I'm very glad that so many young people came, and I'm sure that even more are trying to follow what's going on online. My name is Jan Kopecký. I am responsible for the leadership of this institute, and I will show you some introductory slides before you will be exposed to the real science. So our institute is one of the largest biomedical institutes of the academy. You see that we have about 400 employees. Most of them are scientists, and among the scientists, we also count our PhD students who are very much involved in what we are doing. So we have more than 20 scientific laboratories, as you might know from studying our website. The origins of the institute go back some 65, 70 years ago. We were just discussing what is the anniversary this year. And the institute was founded by a couple of very important scientists. All of them were medical doctors, and they started the tradition of the research that we are still continuing here. So neurophysiology, muscle, heart, ontogenesis, medical research oriented on prevention of major diseases. And still we have these three major fields of interest: neurophysiology, cardiovascular physiology, and specific areas of metabolism. So this is what you will be able to study here, or to help us to do here. So we have three major systems, which is the central nervous system, we have cardiovascular system, and we have metabolism. And when we are trying to characterize the basic mechanisms, we are very much using laboratory animals, also in vitro system. And then when we would like to verify what is going on in the animals, we have collaboration with clinical scientists, and we have this area here. So we have clinical studies which we participate in, and we have our animal models and in vitro models to study basic biological mechanisms with the aim to improve prevention, diagnosis, and treatment of major non-communicable diseases, namely lifestyle diseases. I will show you in a minute what does it mean, lifestyle diseases. And we have also specific interests in ontogenesis, how the system develops, how the development of specific diseases depend on age and things like that. So this is very important aspects of what we are doing here in the Institute of Physiology, because ontogenesis is something which is very dynamic, and changes are occurring during ontogenesis. Everything must be under control. So this is also very interesting going on during the development, prenatally, postnatally, aging. Okay. And what are the lifestyle diseases? So we know that obesity is one of the major troubles in our society, including Czech Republic. And obesity itself is not a disease, but it's a predisposition to real diseases. And obesity is associated with diabetes, there is a very strong association, and also with some other diseases like disease state like hypertension, dyslipidemia. And if you have a diabetic patient, the chances that it will suffer from cardiovascular diseases are about fourfold higher compared to a lean person. And if you are a diabetic patient, you suffer from cardiac disease, then your chances that you will die by due to the cardiovascular diseases are even higher than in the normal population. But as a matter of fact, more than half of us will die eventually from cardiovascular disease. And this is true. And this is why we are trying to study these basic mechanisms here. And this is very expensive to take care about disease patients here, about the patients. So more than 25% of expenses from the healthcare system goes to the treatment of complications of diabetes only. You can make this judgment. And we are also interested here in the diseases of neural system. And even here we have a strong link because diabetic patients have a very high propensity to develop Alzheimer's disease. Alzheimer's disease, is something which is dependent on age. So if you are old, your chances to get Alzheimer's disease, unfortunately, are also very high. And if you are a diabetic patient, you have a lot of troubles, including neuropathic pain. Dr. Palecek will tell you something more about this. So everything is interconnected here. And even COVID pandemics, was affected by our predisposition to these diseases, and whether you were diabetic or obese patient or not. So you learned during last years that even this is important here. And why these diseases are interconnected? Because genes, environment, age, all these factors count in development of these diseases. Doesn't matter whether this is diabetes or cardiovascular disease. And to study the diseases or the mechanisms, you must have special tools, and you must have clinical patients available, laboratory rodents, ex vivo systems, and you also like to study things at the molecular level. And you study different mechanisms, which are all of them are involved again in development of the diseases I was talking about. So you have inflammation, energy metabolism, circadian rhythms, and so on. And to do real research, which leads to some results, you need a lot of different methods, because these diseases are complex. The factors which are involved are multiple. And to characterize the factors and mechanisms, you must have a lot of different methods available. And this is something which we are trying to develop systematically at the Institute. So we have so-called phenotyping at the whole organism level. I will show what does it mean. Then we have metabolomics, proteomics, transcriptomics, and also we would like to know more about the genetic background. So you must have access to all of these methods to be able to do some real improvement in here. So when we are talking about phenotyping at the whole organism level, we have systems that allow you to study behavior. We have a system that is able to characterize metabolism at the whole body level. We can do the various type of whole body imaging to show liver or heart or adipose tissue in the body. And then we have methods that allow you to study organs and also characterize ex vivo metabolism of tissues. And then you go even deeper to the level of individual analytes. And we have now a very strong facility here focused on metabolomics, which is able to really identify all the molecules you can imagine, but then you have a lot of information, and which is not mentioned here. when you get a lot of information, you must be able to somehow to understand all the data you have. So you have so-called bioinformatics, you know. So this is something which is developing here also, and this is the very difficult part. And what you need in our institute, because we do a lot of studies using animal models, you need animal facility which allow you to do the experiments. So we were lucky that these buildings from 60s from the last century are now under reconstruction, and we are at the end of this reconstruction. So by the end of this year, we should have very modern animal facility, which will allow you, or maybe of course my colleagues now, to perform studies on the animals under the conditions which are appropriate, you know, which is very important. We don't want animals to suffer, but we need them. We need to do animal experiments under appropriate conditions. Now we have it, 200 million Czech crowns now, and you know, well, that nowadays it's very difficult to finance basic research, and we are lucky at this institute because now we are supported by two major grants which we have in collaboration with clinical scientists. And one type of funding is for conducting research in metabolic and cardiovascular field with 5 partners. One of the major partners is Institute of Clinical Experimental Medicine across the street. We have access to patients, and 9 million euros for the institute is not little, right? And we have another type of funding for studies in neuro research. Again, some labs are involved. So in total, more than 70% of our laboratories are now supported by these 2 grants, which is an advantage nowadays because, you know, funding of science is difficult to get. And that's why we need more motivated students coming. We would like to have excellent students coming. We can offer something, but we will also ask them to perform. So that's why maybe you are here to learn what are the possibilities, but this is what we can offer. And just to illustrate what is the situation at the institute regarding PhD studies, we have a lot of diploma students. I don't know how many, but we have about 80 PhD students here at the present. And this is the split in various teams of the PhD studies performed during the last 5 years at the institute, and we are proud to say that more than 80% of the PhD students were able to finish their PhD thesis here. So this is a good place for doing your studies.
Thank you very much for the introduction, and I will continue with few slides concerning the our students, what we offer for students. Just this to share the screen also with online with people which are connected online, and currently there is open a call for applications for PhD studies. The deadline is 20th of March, and there is an online application form available on our webpage. And there is also the full list of available positions there where you can find what you are interested. You can choose up to 3 different topics that you can apply for your PhD study. For PhD students, we offer several benefits. As Mr. Director said, we have research in the fields of neuroscience, cardiovascular physiology, and metabolism. We use a large amount of a lot of methods, now current methods concerning physiological, biochemical, and molecular biology. You can get also employment up to the full-time salary and benefits depending on the budget of particular laboratory. And we organize regularly several events for PhD students, like seminars, course of physiological methods, or every 2 years students meet at their own conference. There is an advancement report in the third year of your PhD study that is just to check if your PhD study proceeds well and you are able to defend in the next year. Then we offer also the internal PhD thesis evaluation that you can train before your final defense. You can train your defense of your thesis here at the institute. You can follow some English language courses. And what is also nice that directly in the campus, we have on-site accommodation in this natural environment. There is a lot of forest animals around and also some sport facilities. So this is as concerned PhD program. And for bachelor and master science programs, there is also a list of available topics at our webpage. And in this case, I recommend to contact particular supervisors, which is named at the topic, and ask him if the position is still available, and you can discuss it directly with him or her. And now, how will be organized this day? We will have a morning session with presentations of 12 laboratories, and afternoon, you are here on site, you will have a possibility to visit up to 3 laboratories according to your choice. And for the registration to the visits, you register at the in foyer. There are some papers and you can write your name at particular times. The visits will start at 12:30, 1:15, and at 2 p.m. And now I wish you enjoy the day. And we are a little bit in delay, so I will ask Martin. I will ask Jiří Paleček to introduce his topic for bachelor, master science, and PhD study. He's the head of the Laboratory of Pain Research.
So, good morning again, and welcome to the world of pain. I hope you'll enjoy it for the last 10 minutes. And as was said, yes, wonderful, thank you. I will tell you something about what we do in our department, the laboratory. And if you are talking about pain, you probably would understand that acute pain is an important physiological mechanism that prevents injury in the organisms. However, chronic pain is not really that advantageous to the patients and to you. And it's really a pathological state which has very strong negative impact on the patients. We have different types of pain. And while probably all of you know about the nociceptive pain, which when you, let's say, burn your finger and make some injury, there are also types of neuropathic pain, which are coming from the damage of peripheral central nervous system, which could be induced by traumatic injury, diabetic neuropathy, chemotherapy, treatment, and some other diseases. So it's important to know that really about 20% of people with diabetes have neuropathic pain. Also, a high number of people with cancer treatment have neuropathic pain. And we have really very limited possibilities how to treat the pain in these patients. And it's also because the mechanisms of neuropathic pain are different from the mechanism of acute pain. And therefore, the classical analgesic drugs like opioids and ibuprofen and acylpirine and whatever doesn't really work. And we have to find new drugs to treat these patients. So there is really a high unmet need for new analgesic drugs with minimal side effects. So when we study pain, of course, we know a lot today about the pain pathways and how it develops. But we don't have time for that. But we can generally target pain in either in the periphery, the nociceptors, or we can target the synaptic transmission, especially at the spinal cord level, or we can target the areas in the brain which are involved in pain perception. So in my laboratory, we are concentrated on the possible treatments at the spinal cord level, modulating synaptic transmission at this level. So what do we do really? We are involved in several projects. Some of them are placed in here. So we are interested in chemotherapy-induced neuropathic pain, in the neuroinflammatory mechanisms at the spinal cord and peripheral levels, which are leading to chronic neuropathic pain syndromes, as was mentioned diabetic neuropathic pain, and also in the role of special receptors. One of them are TRPV1 receptors, which may be known also as a capsaicin receptors. You might have heard about them, activated by the chili peppers, but they are very important for pain perception in many levels. About the inhibitory controls by GABA receptors, and also in the interaction of opioid, cannabinoid receptors, and TRPV1 receptors in this respect. Besides this kind of basic research, we have also a translational research, which is in cooperation with the Institute of Organic Chemistry and Dr. Kudova. And there we are really testing new steroidal compounds for possible treatment of pain, and we are developing new drugs, and hopefully we are moving in a good direction. We have some nice results with that. We are using different types of methods. One of our basic methods is electrophysiology. We are recording with patch clamp technique from individual neurons, from spinal cord slices, DRG cultured neurons. And we are also moving to develop cell lines of human DRG neurons, because we know that the transmission from the rat to human is quite far away. And it's important for us to move to some human studies. We are using optogenetic activation of neurons by light, calcium imaging, behavioral tests, and classical immunohistochemistry, biochemical, and PCR, and other methodology. So just to give you a hint, what we do, I will just show you a little bit of our results from the chemotherapy-induced peripheral neuropathy and pain, which is really induced in patients which are treated, for instance, by paclitaxel, which is a drug to treat cancer like ovary cancer, breast cancer, and others. And these patients will develop, about 30% of them will develop strong neuropathic pain conditions, either during the treatment or when the treatment is finished. And it's very difficult to treat. So we are looking for the mechanisms and possible new drugs that could help them. And we are, as I said, concentrating on the role of modulation of synaptic transmission at the spinal cord level and the involvement of different receptors in this modulation, which leads to the pathological pain. So in this respect, we published a number of papers which are quite cited and show that the toll-like receptors 4, TRPV1 receptors, and different kinases play an important role in this respect. And the last paper I will like to show you just in another 1-2 minutes what type of experiments we do. So for instance, here you can see the results of the behavioral experiments, where we can see that after paclitaxel treatment, at increased sensitivity to mechanical stimuli, pain will develop, which in this case was prevented by inhibitor of phosphoinositide 3 kinase inhibitor, which is used for treatment of hematological disease, in fact. We are using electrophysiological approach, recording patch clamp of individual neurons in spinal cord dorsal neurons. You can see in this case, there is a high increase of activity of TRPV1 receptors here invoked by capsaicin, which was prevented by the drug. We are using optogenic activation here. There was a flash of 5 milliseconds of blue light activating inhibitory currents in spinal cord neurons, which was diminished by the paclitaxel treatment, and we could prevent it by this inhibitor. And we are using immunohistochemistry analysis, in this case of dorsal root ganglion neurons, and the expression of phosphoact activation in this disease. So these are kind of the approaches we are using. And we have a nice team, friendly group, I think. We have a lot of fun with our research. And we are looking for pre-graduate students. And we can, for those who are really interested in the work, we can offer some financial support for the work. And we are looking also for 1 or 2 PhD students for the future. So that's probably all from me when the time I was allotted. And those of you who are interested who can come in the afternoon and talk to us about what might interest you. Thank you.
Thank you very much, Jirka. And the next presentation will be the Laboratory of Cellular Neurophysiology, and Teresa Smejkalova will tell us more about the topic from her research.
Thank you. So I will speak for the Laboratory of Cellular Neurophysiology. Lately, we've been particularly interested in neurodevelopmental disorders, which unfortunately typically are lifelong conditions. So they place a significant burden on the patients and their families and on society. And they are complex disorders with polygenic origin generally, but it is clear that some form of dysfunction of glutamatergic signaling contributes to all of these disorders. Our group has for a long time been interested in glutamate receptors, especially ionotropic glutamate receptors in the synapse, and specifically NMDA receptors. We know, it is well known, that NMDA receptors are important for synaptic transmission at excitatory synapses. They're important for synaptic plasticity, and they play a role in development. So it is not surprising, or it makes sense that variations in NMDA receptor genes, the GRIN genes, may be associated with some neurodevelopmental disorders. So we sort of look at this question at many levels. We do some genetic studies where we hope to identify new genetic variants that can be linked to a higher risk of certain neurodevelopmental conditions. And we also work to characterize the gene variants that have already been identified, discovered. And so we do that by looking at receptor function, protein function, and then also at the properties of synapses and neuronal networks. And we also look at animal behavior in our mouse and zebrafish transgenic animal models. And I'll just give you one example of sort of what a project like this might look like. So this figure down here shows a partial structure of the NMDA receptor complex. Here is its transmembrane domain, enlarged here. And in red are highlighted amino acid residues that have been found to be altered in individual human patients with neurodevelopmental disorders like intellectual disability or epilepsy or autism spectrum disorders or schizophrenia. There's actually a significant gap currently between the genetics information, so we can sequence genes a lot and we can get a lot of data that way. So we know about 700 of these spontaneous de novo missense or deletion mutations that alter the protein structure have been identified from human patients, about 700. And probably less than 10 of them have been studied in animal models. It's sort of, it takes time to get to that stage. So we're hoping to fill that gap and to really learn what a single amino acid substitution in a protein, what it would mean functionally. So to do that, we usually start with heterologous expression systems. So we can express our mutated NMDA receptor subunits in some non-neuronal cells, such as HEC cells. And then we use patch clamp electrophysiology and pharmacology to characterize the properties of these mutated receptors. And so in these cells, we activate the receptors by applying the agonist, glutamate, and then we record these electric currents flowing through the channels, through the receptor. And we can also use different drugs, modulators of NMDA receptors, to study how the receptors behave and compare the wild type receptor responses with responses of receptors where one of the subunits has a mutation that comes from some patient. So we've done some work like this, but this is not enough because these are synaptic proteins. They are neuronal proteins. So to really understand what a mutation in this protein would do, we need to look at neuronal function. So to do that, we prepare neuronal cultures from our transgenic mice carrying a specific human patient-derived mutation. And then we, again, do patch clamp electrophysiology as well as biochemistry, immunofluorescence, et cetera, to characterize these receptors in neurons in their more native context. And we study the properties of synaptic transmission in the presence of a mutated NMDA receptor subunit. And any changes in synaptic transmission in turn are likely to lead to larger scale changes in the behavior of neuronal networks. So we are also studying circuit function by using live cell calcium imaging, using the GCAMP indicator in cultures, again, prepared from mice with these mutations, to study the activity in the circuit, the properties of the circuit, and its development over time. And we can also, in all of these cases, test different pharmacological modulators with the hope of finding some treatments for these patients. And finally, in collaboration with the Czech Center for Phenogenomics, we do detailed behavioral characterization of these GMO animals. So we look at their general activity levels, their anxiety, their cognitive function, their social behavior, and more. And again, the goal of all this is to better understand how these gene variants in NMDA receptor subunit genes may lead to disease, complex disease such as autism or schizophrenia, and to potentially identify any treatments available for these patients. So this was us a few years ago. So we need to take a new picture soon, and maybe you'll be in it. And yeah, come talk to us in the afternoon.
Thank you very much, Teresa. And next presentation will be Laboratory of Neurochemistry. First speaker will be Alena Randáková and second Helena Janičková, and they will present the topics for Bachelor, Master Science, and PhD studies.
Good morning, everyone. My name is Alena Randáková, and I'm from the Laboratory of Neurochemistry, and it's my pleasure to present here the research topic focusing on our laboratory. So the topic is the mechanism of selective activation of individual subtypes of muscarinic receptors. Muscarinic receptors are membrane proteins, and they belong to the G protein coupled receptors that transmit the signal into the cell through heterotrimeric GTP binding G proteins. We have 5 subtypes of muscarinic receptors, M1 to M5. And depending on subtype and localization, muscarinic receptors mediate variety of physiological function of their natural agonist acetylcholine, ranging from a complex of higher nervous function like learning, memory, mediated mainly by M1 receptor, to vegetative processes, for example, like smooth muscle contraction mediated mainly by M3 and many others. Muscarinic receptors present a promising pharmacological target in many pathological conditions. On this slide, you can see some pathological conditions where inhibition or activation of individual subtypes offer interesting pharmacological or provide pharmacological opportunity. Because of a variety of physiological function mediated by individual subtypes of muscarinic receptors, it's very important to modulate individual subtypes selectively. But it's quite challenging because the orthosteric binding site, this binding site for acetylcholine and other muscarinic ligands, is very conserved among individual subtypes of muscarinic receptors. On the other hand, individual subtypes of muscarinic receptors differ in signaling profile. While subtypes M2 and M4 preferentially activate GIO G proteins that leads to inhibition of adenyl cyclase and decreasing of cyclic AMP, subtypes M1, 3, and 5 prefer coupling with GQ11 class of G proteins, leading to accumulation of inositol phosphates and calcium release. So we believe that targeting to the particular signaling pathway can lead to selective activation of individual subtypes. And how we can do it? Muscarinic receptors are very flexible, as other GPCR, are very flexible structures with a flexible G protein interface. And the muscarinic receptors are able to interact with the individual isoforms of G proteins with different potency, efficacy, and kinetics. And they are also able to activate non-preferential G proteins. And biased agonists are a type of agonist that structurally different agonists are able to induce the agonist-specific conformation that can lead to non-uniform modulation of signaling pathway. So biased agonists can be specific to the particular signaling pathway. And our project focusing on analysis of signaling of novel muscarinic agonists. We will analyze the direct coupling with the individual G proteins and their isoforms. And then we will also analyze the second messenger system. This is what we offer in our project. So that's all for me, and now Helena.
Now Helena Jančková from the same laboratory concerning muscarinic receptors.
So good morning, my name is Helena Jančková. I'm from the same department of neurochemistry, but I will be presenting a completely different project. It's actually not on muscarinic receptors, but on nicotinic receptors. The project is titled Significance of Nicotinic Acetylcholine Receptors Expressed by Specific Neuron Populations in the Striatum. And since I don't want to lose anybody at the very beginning, I would like to go quickly through the keywords. So the nicotinic acetylcholine receptors, they are receptors for acetylcholine. They are ionic channels widely expressed throughout the brain, and they are responsible for many cool behaviors, including smoking, attention, or anxiety. And the striatum is a brain structure important for many, many things, but maybe it's most infamous for its connections to various neurological disorders, including Parkinson's disease, Huntington's disease, or addiction. And in the striatum, there is a rich variety of different neuron populations. And these different populations are very diverse, differing in terms of their appearance, connectivity, firing patterns, or gene expression. And basically the main question of the PhD project you should be investigating is how the nicotinic signaling controls these specific neuronal populations in the striatum, and through this control, how it reflects, how it changes the behavior in mice. Why is it even worth it to study a thing like that? Is it because the striatum is a big hub for behavior in mice and in humans as well? Recently in a scientific publication, I saw a slightly unscientific claim. They were saying that approximately 90% of behavior is passing through the striatum. But, you know, unscientific as it may sound, it might be actually true. And because the striatum is so important, it's very important to understand better how it works, and even more importantly, to be able to control how it works. And you know, in neuroscience, a lot of people are using different tools, how to control brain structures. Many people are using optogenetics or chemogenetics, but we believe that maybe there is more to neurons than being just on and off with optogenetics, and the neuromodulatory receptors such as nicotinic receptors can offer a finer tool for this modulation. So now I would like to spend just a couple of slides showing you what is the project is actually about and what has been already done. So in literature, there has been long suspected that nicotinic receptors or the expression of nicotinic receptors in striatal neurons is actually very limited. And this knowledge was based on electrophysiological experiments mostly, because for the lack of antibodies working in the brain tissue, it's impossible to image most of the nicotinic receptors by immunohistochemistry or immunofluorescence. And so we were able to confirm this claim recently by fluorescence in situ hybridization. and we saw that the most common nicotinic subunit beta-2 is actually expressed mostly by cholinergic interneurons in the striatum, which is a very rare, not very numerous population of neurons. And 80% of beta-2 subunit was expressed by these neurons, with more minor contribution by parvalbumin positive interneurons and neuropeptide Y positive interneurons. So when we saw this, we decided to delete this beta-2 nicotinic deletion from this rare neuronal population and see if there is any effect on behavior. And for this mutation, we use two different approaches: Krelok-Spi approach that allows us to do less specific, maybe more powerful, more prevalent deletion, and CRISPR-based approach that allows us to focus only on a specific population of neurons. And we decided to choose, we chose the NPY expressing interneurons in the striatum. After the deletion of the beta-2 subunit, we tested the mutated mice in different behavioral tasks. It was really long batteries of tasks we subjected these mice to, but in the end, it appeared we really didn't need to bother so much, because the only, well, not only, but most of the positive effects we saw in our mice were only in two behavioral domains. It was social behavior and anxiety-like behavior. On the other hand, the character or the valence even of these changes were quite different, depending on which neurons we focused on, which neurons were subjected to the deletion, or which structure we focused on, because we also moved outside the striatum and performed the deletion in the prefrontal cortex. So here are just a few examples. On the left side, you can see the deletion using the Krelok-Spi system. In the striatal neurons, the deletion of beta-2 is inducing a decrease of social behavior, decrease of sociability, and also increase of anxiety. When we focus only on NPY expressing interneurons in the striatum and perform the deletion in these neurons, we saw affect impairment of social memory and again increase in anxiety. When we moved outside the striatum to the prefrontal cortex and performed a specific deletion of beta-2 nicotinic subunit in NPY positive neurons in the prefrontal cortex, we actually saw the opposite. We saw the increase in sociability and decrease in anxiety. Of course, it's very nice, or it would be very nice, to associate these behavior changes with changes in neuronal activity, but unfortunately we are no electrophysiologists, so we had to circumvent this problem. We decided to measure CFOS as a proxy for neuronal activity, and we were able to show in our mice that the deletion of beta-2 nicotinic subunit in the striatum, in general, increasing striatal activity or activity of striatal neurons, increasing CFOS expression in the striatum. But when we were focusing only on cholinergic interneurons and measured the FOS expression in cholinergic interneurons only, the FOS expression were actually decreased, which is in line with the fact that these interneurons are the only, or not the only, but the main population expressing this beta-2 subunit. So they are not as much activated when we delete it. And now finally, I would like to get to the point of this presentation. What is the next step? What is the goal of the PhD project we are offering now? So it's actually to stop circumventing the problem of neuronal activity and to measure directly changes in neuronal activity, in neuronal firing or properties in the striatum after the beta-2 deletion. So still we are no electrophysiologists. So for this, we established a collaboration with a laboratory in Belgium that has extensive experience in electrophysiological recordings in the striatum. And it means also that at least 6 months of the PhD will be spent in this Belgium laboratory. In a nutshell, the main goals of the proposed PhD project are as follows. So first, basically repeat what we already did in focusing on slightly different cells. So doing the beta-2 nicotinic deletion in cholinergic interneurons using the Shed-Cree mice and Krelok-Spi or CRISPR-based approach, and determine behavior changes induced by this deletion. Second, to move to this Brussels laboratory and measure the effect on of this deletion on neuronal activity in the striatum. And lastly, determine effects on physiological or on activity of neurons using calcium imaging, which will be done again in Prague. That's it, I believe. Just I would like to mention that if you are not ready, if there is anybody in the audience who is not ready for PhD yet, we also offer related projects or topics for bachelor and master students. So don't be afraid to get in touch. And that's it. Thank you for your attention. Maybe see you in the afternoon.
Thank you both speakers from the Laboratory of Neurochemistry. And the next speaker is Martin Balaštík from the Laboratory of Molecular Neurobiology.
Okay, good morning, everybody. My name is Martin Balaštík, and I'm the head of the Laboratory of Molecular Neurobiology here at the Institute of Physiology. And in the next couple of minutes, I would like to introduce you my laboratory, the research we are doing, the topics, the questions we are interested in, and the models and methods we use to, in trying to address them. Now, we're going to be talking about microtubule-associated proteins and their role in neural development and disease. Now, why is that important? As you probably know, or you may know, in the human brain, there are considered almost 100 billion neurons present, forming 10 to the 14 synapses, which is number of synapses or connections that you would have if all the people in the planet and 10 other planets would get connected, 10 or 1,000 connections per person, and then you just wrap it in a small little package that is content of your head. And at the same time, those connections are so precise that neuron from one part of the brain, for instance, from thalamus, is projecting to another region, but not only to the specific region of cortex, but also to a specific cortical layer and forming their connections with specific neurons. How is this precision achieved is a very important question, yet unanswered question, but it's clear that development of brain is a mechanical process. And all its important steps, which is proliferation of the progenitors, their migration, formation of connectivity and differentiation, is a mechanical process and requires some motor system that is allowing it to proceed. And obviously, this is allowed by cytoskeleton, neuronal cytoskeleton, and its integral part, which are microtubules. And that's why if you look at what happens when you have some mutations or changes in microtubules or the whole cytoskeletal system, you will see that in a number of neurodevelopmental disorders like lissencephaly, heterotopia, microcephaly, genes that are either tubulin isoforms or microtubule-associated proteins are mutated in them and were associated with pathogenesis of those neurodevelopmental or neurodevelopmental disorders, or those are cortical malformations. Now, what are those microtubule-associated proteins? Because this is something that we are interested in. Those are intracellular proteins. They are interacting with microtubules. They help to organize microtubule cytoskeleton. Their function is really numerous. They have numerous functions, but critically in neurons, they regulate microtubule-based transport and microtubule dynamics. And this is something that could be said about one group of proteins that we are interested in, or lately we were analyzing a lot, which is collapsin response mediator proteins, or CRMP2. Those are binding to microtubules, regulating the microtubule-based transport as well as microtubule dynamics. And they were linked to pathogenesis of schizophrenia or autism spectrum disorders. Also, they are hyperphosphorylated in Alzheimer's disease. And in my lab, we try to analyze and address the question what the changes of this group of microtubule-associated proteins and its changes by alternative splicing by activation various signaling cascades, phosphorylation, conformational changes, how they change this microtubule-associated proteins, and what is the effect of those changes on microtubule dynamics, on axon guidance, synapse formation, and cortical development, or versus neurodevelopmental disorders. Now, what kind of methods we use in my laboratory to address those questions? We are generating knockout mice. We have generated already 2 knockout lines, full CREM2 knockout mice, and isoform-specific CREM2 knockout mice that have only one of the isoforms missing. We use in utero electroporations that allows us to modify only some specific part of the developing and postnatal nervous system, where we inject into the brain some plasmids, or the DNA that we are gene that we are interested in, and after electroporation get a specific expression only in part of only in the targeting neurons, which you see here, and we can analyze exactly what this expression or downregulation of a specific gene affects, how it affects growth of the projections. Here you see it in the cortical, in the section of the coronal section of the cortex, how the projections look, how they target their cellular targets, and if there are any defects in pathfinding of those neurons. We also using organotypic ex vivo cultures, we study migration of somehow either upregulated or downregulated microtubule-associated proteins, how they affect migration of the cortical neurons, as you can see here. We use fluorescent labeling to analyze projections of some specific neurons, and how mutations in microtubule-associated proteins affect pathfinding really in vivo using various mouse models. We use gene gun labeling of neural brain slices, where we can randomly target and analyze in detail the whole cytoarchitecture of neurons that have been somehow either mutated, upregulated, downregulated, some of the microtubule-associated proteins we are interested in. We use primary neuron cultures or explant cultures that allows us to study really specifically some details of neuronal growth and the effect of microtubule-associated proteins on axons, or for instance here, growth cones, as you can see here. We are using microfluidic chambers that kind of is separating the neurons into 2 compartments, the cell bodies as well as axons, and we can separately analyze how some changes that we inflict in them, or some extracellular signaling or stimulation affects growth of the axons in vitro. In collaboration with Institute of Biotechnology, we are analyzing how microtubule-associated proteins affect directly polymerization of microtubules. As you can see here, those are in vitro pieces of microtubules, and microtubule-associated proteins here of the CREM2 family that are promoting the polymerization, very dynamic polymerization of microtubules, and we can really analyze it directly in vitro. And we use multiple of really advanced microscopic techniques, including confocal microscopy, TIRF microscopy, light sheet microscopy, to analyze really details of the formation of neuronal connectivity during the development, and the effect this would have on the whole even behavioral and physiological conditions of those mice that we generate. These are some recent publications that are related to the work that we have done in the lab. You can find them online, and this is my laboratory. I have to thank them for the great work they do in analyzing the mice that we are generating, and you know, all the other questions, as well as the various collaborations that we have both in Prague as well as in international laboratories like in Institute Curie. And this is all from me for today. Thank you for the attention. If you are interested in similar topics, you can ask me, you can contact me, or you can just step by today in the afternoon and we can discuss what we can do. Okay, thank you.
Thank you very much, Martin. And now we will move to the cardiovascular research from neurophysiology. And I would like to welcome here Ivana Vaničková, and she is the head of the department or laboratory of experimental hypertension.
So, as was already stated, I am the head of the laboratory of experimental hypertension. Hypertension is, hypertension is a very important disease which affects many tissues and organs. So it causes deterioration to vessels, causes atherosclerosis, causes stroke to the brain, retinopathy in the eye, or renal or cardiac deterioration. Therefore, its treatment is very important. Our laboratory is focused not only on basic research, which is on the left shown here. We study central and peripheral mechanism of blood pressure control, as well the role of oxidative stress and mitochondria in these processes, as well as the role of autonomic nervous system. We also have the part of our research devoted to the biomedical research. That means that we are studying the effects of different drugs in hypertension treatment, and our colleagues are also connected to the Institute of Organic Chemistry and Biochemistry, studying the role of newly developed neuropeptides in obesity. Our research is based on the rat models of hypertension, which are both the genetic models of hypertension. The mostly used model is spontaneously hypertensive rats, which is the rat model, which is defined as with the activated sympathetic nervous system. There is also a specific rat model which is sensitive to the increased salt intake in their diet. And we also have here a unique model of hypertension, which is associated with the activation of renin-angiotensin system. This means REN2 transgenic rats. We are also able to induce experimentally hypertension by reduction of renal mass or renovascular hypertension. And as you can see on the next slide, the two possibilities of this procedure is to remove one kidney entirely and two poles of the remnant kidney, or the placement of the silver clamp on one renal artery. These models in the established phase are defined as the models of chronic kidney disease. We also have the models for heart failure, which are the spontaneously hypertensive rats in which high fat intake is given, or the model, which I also already mentioned, the dual salt-sensitive rat on high salt intake. Concerning our methodological approaches, we work from the level of a whole animal up to the molecular biology. And the basic of our research is connected with blood pressure measurement, of course. So we either use the long-term or chronic blood pressure monitoring via telemetry, or the chronic measurement with tail cuff. You will see this in the afternoon session, experimental. We also work with cannulated conscious rats using this approach as a acute for the acute experiments. And we are also able to place the cannulas to the lateral brain ventricles, and we are able to apply various drugs to this part of the brain and see how it affects the blood pressure. We are also able to work with perfused vascular beds to have the signals from the body, or to work on the lower level with the isolated arteries. So it uses the wire myography techniques. Our methods also contain basic biochemistry, immunohistochemistry, and also, as I have mentioned, also molecular biology. So our PhD project is the gliflozin treatment in the treatment of non-diabetic models of hypertension and heart failure. Sorry, it works somehow strangely. Gliflozins are primarily anti-diabetics, and they work through the increase of glycosuria and sodium, so the inhibition of sodium glucose transporter. And thus they produce higher natriuresis and glycosuria, and thus they decrease body fat mass and also proteinuria. Through the increase in excretion of sodium, they also decrease blood pressure, and their effects was also described beneficial for the heart. So all in all, they cause renoprotection and cardioprotection in the human studies. And these are the most important clinical trials which were until now done in humans. And these first studies demonstrated that their beneficial effects are positive in diabetic patients. But later on, it was also described that the beneficial effects are present also in non-diabetic patients. And moreover, also in patients with chronic kidney disease and heart failure. And the question is, what are the mechanisms which are beyond the anti-diabetic effects? And therefore, we perform several studies in 3 different rat models, ran-2 transgenic, hypertriglyceridemic rats, and spontaneously hypertensive rats. And all these beneficial effects were connected with decrease of body weight and visceral mass. But depending on the rat models, there were also additional other effects concerning blood pressure decrease in enterocyte transgenic rats, improvement in hepatic lipid metabolism, and inflammation and oxidative stress in spontaneously hypertensive rats. So it means that there are specific effects which are seen beyond the classical anti-diabetic effects of these drugs. And what we like to study is the effects in heart failure and hypertension. So our laboratory collaborates with laboratories inside of this institute and also outside, and we have also international collaboration. As you can see, sorry, it works so strangely today. So you can see that our team spirit is seen not only in our laboratory, but also outside of our laboratory. And if you are interested in glyphosines, please contact me and we will see maybe in the afternoon. Thank you.
Thank you, Ivana. And we are like 3 minutes delay, and now we will have here, we have prepared a coffee break for you to take some rest, and we will be back here at 10:30. Yeah. So for those that are online, please make your own coffee and come back at 10:30. Yeah. Break. I hope that also those that are connected online are again online. And we will continue our presentations of topics that are available for students here at the Institute of Physiology. And the first speaker will be Hanna Kubová from the Laboratory of Developmental Epileptology. So we are going back to the neuroscience for one presentation.
Thank you. So welcome in the Institute of Physiology. I would like to present you one of our research topics. As you already heard, I am a head of department of our Laboratory of Developmental Epileptology, and our major or our research topics are closely related to brain development, long-term effects of early brain injury, and also to development of safe medication for pediatric use and testing safety of drugs that are used mostly to treat epilepsy. So mám to moc nereagovalo teda. Jo, dobrý, slabý prstíčky. So the brain continues to develop long term after we are born, and brain development is basically driven by interaction between biological and environmental factors. Environmental factors, by this we mean all possible chemical or physical interventions brain can be exposed. Neural activity is critical for brain development, and neural activity is controlled by neurochemical systems, by brain chemistry. What is important, because all these neurotransmitter or neuromodulatory systems are keeping balance between excitation and inhibition in the brain. And any interruption, this very fine balance that is changing during development, because at early stages of postnatal development, excitation is predominating. This maturation, inhibition takes over. So any interruption of this process can affect brain development and later brain functions. What is important to say that most of neuroactive drugs we are using to treat pediatric patients, I'm not talking about illegally used drugs, but about drugs that are commonly and frequently used as anti-epileptic or anti-seizure drugs, if you will, anesthetics, drugs inducing sedation, or anxiolytic drugs are acting through their interaction with brain chemistry, with neurotransmitter systems. So $1 million question, are these drugs that are used for tens of years safe for immature brain? Are we putting immature brain into risk when we are using these drugs? This is a really important question because unfortunately drugs are developed during this preclinical testing and also at the beginning of clinical studies, mostly for needs of adult brain. And development of drugs for specific pediatric use or even testing their safety for developing brains are more than limited. Indeed, consequences of early life exposure to these neuroactive drugs are changing during postnatal development because there are so-called critical developmental periods of increased sensitivity to environmental environmental factors. And indeed, basically anything what is happening during these critical periods has strong effects on brain development and functions. And what happens, or alterations that are somehow induced during these periods, are not repellable during later developmental stages. So basically, we expect that effects of drug exposure during these critical developmental periods are negative. And there are really a lot of data, preclinical data indeed, showing that exposure, even short-lasting exposure, especially in rodents, to, again, commonly used drugs, certain anti-epileptic drugs or anesthetics, can permanently affect brain morphology, structure, and also brain functions, and can somehow increase the risk of development of some psychopathologies or behavioral abnormalities later in life. On the other hand, there are also data showing that under certain conditions that really have to be defined precisely, early drug exposure can have positive long-lasting effects, and it can even prevent or favorably modify development of some neuropsychiatric diseases in patients in risk. It's necessary to point out that most of drugs that are used in pediatric practice exhibit the same immediate effects in both adults and children. So anti-epileptic drugs are preventing seizure development. Sedative drugs are inducing sedation in neonates, in adults, in young adults, in elderly people. But major difference is that whether whereas in adults, drugs or drug effect subside usually shortly after the end of therapy, situation in immature brain can be quite different, and drug effects can somehow secretly incubate, and we can see changes in brain functions long time after cessation of drug exposure. Indeed, this is something what is very difficult or even impossible to study in humans. On the other hand, we have such a tools in laboratory animals because we can precisely control conditions. We can use healthy, naive, immature animals, as well as animals that are surviving some kind of brain injury or brain insults. So we can make some kind of picture, and we can identify not only consequences of this treatment, but also mechanisms that are responsible for alterations of brain functions. This is just one example from our laboratory. We used basically the most commonly used neuroactive drugs, benzodiazepines, and we exposed neonatal rats just for 5 days to clinically or pharmacologically relevant doses. And we found that even such a short exposure is changing social behavior and social skills of these animals for the rest of their life. And this is not only change that can be identified, and benzodiazepines that are, by the way, used in many indications in all age groups, including neonates or preterm babies, are not only drugs with this kind of negative effects. So again, aim of our research is, one of aims, is to determine long-term impacts of neuroactive medication on brain development. And again, we are mostly focusing on anti-seizure drugs and epilepsy per se, and also to identify mechanisms that are responsible for these changes in order to help to develop more safe drugs for pediatric use. We are using multidisciplinary approaches, behavioral studies. We have a huge portfolio of behavioral tests for all age groups of animals, neuropathology, electrophysiology, and also methods of molecular biology. Unfortunately, I cannot offer you any excursion to our laboratories today because our laboratory is under reconstruction. But in a case you are interested in this topic, please let me know. You can visit us. We can talk in more details about what we are doing and what we can offer to you. Many thanks for attention.
Thank you. And next speaker will be Petr Zohar from the Laboratory of Adipose Tissue Biology, and he will present two topics for PhD studies and master science.
Okay, hello everyone. I would like to introduce our Laboratory of Adipose Tissue Biology and to tell you a few words about PhD projects we are now hiring for. So we are like a middle-sized group of researchers. We have currently 6 PhD students. We are looking for 2 more. 2 our PhD students are Czechs. We have also 2 Serbians, 1 Indian and 1 Nigerian student. We are interested in anything which has some connection with obesity and associated disorders like type 2 diabetes. We work with a broad range of experimental models, starting with cell cultures like 3T3L1 adipocyte cell line or primary hepatocyte and adipocyte culture. Most of our research is based on work with mice, and we also have collaboration with some clinical institutes and hospitals which generates biopsies and clinical samples which we later analyze. In past, we were focused on several topics or strategies, how to counteract obesity or to improve metabolic health, including fish oil supplementation, calorie restriction. Nowadays, we are interested in exercise and also in cold exposure or acclimation, which I will tell you a bit more now. If you are more interested in our current projects, you can visit our website and find more information there. The PhD projects we are hiring now for are these 2. One of them is rather basic research. The other one is based on our collaboration with Institute of Clinical and Experimental Medicine, and it's therefore like applied or clinical research. Role of non-shivering thermogenesis in cold and obesity resistance. Well, you are probably aware that imbalance in energy intake and energy expenditure can result in accumulation of fat, obesity, connected with some metabolic disorders. Energy expenditure has many components. It's not just physical activity; it's also basal metabolic rate, and quite a big amount of energy can be used just to keep your body in the proper temperature. So if you or a mouse is in so-called thermoneutral environment, the cost for stable body temperature maintenance are covered entirely by your basal metabolism. But if you move to cold, then the cost for thermogenesis rises a lot. So our bodies are ready for this to recruit this energy for thermogenesis, and actually activating these processes could significantly increase the total energy expenditure on the body and eventually counteract the development of obesity. So what are the mechanisms we use to protect ourselves from cold? As you are probably aware, the most basic one is just muscle shivering. And the other classic one, canonical one, is uncoupled respiration in brown adipose tissue especially. What's uncoupled respiration? You are aware that most of ATP in our bodies is produced by electron transport chain in inner mitochondrial membrane, which generates some proton motive force. And when the protons are returning to mitochondrial matrix, they propel activity of ATP synthase. But brown adipose tissue is unique in that sense that it expresses a special uncoupling protein 1, which is a proton channel, providing an alternative way for protons into mitochondrial matrix. No ATP is produced when protons are passing through uncoupling protein 1, and all the energy is dissipated in form of heat. Brown adipose tissue, in contrast to classical white adipose tissue, contains a lot of mitochondria, so it's very efficient organ for heat dissipation. How this process is regulated: if you feel cold, then your brain signals through a sympathetic nervous system from the nerve endings. Norepinephrine is released and activates beta-adrenergic receptors on brown adipocyte. How this thermogenesis or thermogenic mechanism can correspond to obesity. This suggests our research using these models of propensity to obesity. We have two murine strains which differs in the propensity to obesity. If you feed black 6 mice with a high-fat diet, then they develop obesity quite nicely. But AJ mice are resistant to diet-induced obesity. So we exposed these mice either to thermoneutral temperature of 30 degrees, or we kept them for 7 days in cold, and then we study the mechanism of thermogenesis they use. And quite surprisingly, we found out that these AJ mice, in contrast to black 6 mice, has much smaller activity of brown adipose tissue, so the classical organ for non-shivering thermogenesis when they are exposed to cold. So even though both these mice are able to survive in cold to similar extent to protect themselves from cold, black 6 mice relies largely on brown adipose tissue, and AJ mice has to rely on something else. So could this be muscle shivering? No, we didn't find any upregulation of shivering in AJ mice in comparison to black 6 mice, rather the opposite. So neither muscle shivering can explain the lack of brown adipose tissue activity in AJ mice. So there has to be some other alternative mechanism employed in AJ mice. And several such mechanisms were suggested to exist. So we tested several of them. We focused on muscle, skeletal muscle, because besides shivering, there can be some non-shivering thermogenesis existing in muscle, but this is not based on uncoupled respiration. This is based on increase in ATP consumption. So one of very energetically demanding processes in muscle is calcium cycling, because calcium is important for signaling for starting muscle contraction. So there are some calcium pumps on sarcoplasmatic reticulum, and what's interesting, it was suggested that expression of small peptide sarcolipin, if this peptide binds to the calcium pump, then the effectivity of calcium pumping is decreased. So it means that a lot of energy is just wasted and dissipated in form of heat, and it is thought that this mechanism could produce the lacking heat. And indeed, we found that AJ mice, in comparison to black 6 mice, has higher levels of sarcolipin, and these levels further increase by cold acclimation. Other difference between AJ and black 6 mice we studied, or we are going to study within this PhD project, is formation of mitochondrial supercomplexes, because these individual complexes of mitochondrial electron transport chain can work more efficiently if they are in close vicinity, and which provides like more efficient electron flow and can counteract reactive oxygen species production. So formation of these supercomplexes requires some assembly factors. One of them is SCAF1, and black 6 mice are known to lack this SCAF1 protein. So the AJ and black 6 mice are known to differ in the formation of supercomplexes, and we found out that in AJ mice, one particular type of these supercomplexes, I don't see exactly which one, is upregulated in cold. So this could also somehow enable the muscle of in skeletal muscle. This could also enable the muscle of AJ mice to be ready for this calcium cycling and heat dissipation. So AJ and black 6 mice, this resulted in this paper where we are showing that AJ obesity-resistant AJ mice don't use brown adipose tissue to similar extent as black 6 mice, and there are other mechanisms, perhaps in muscle, which can replace the brown adipose tissue activity in AJ mice. So what's now the project for the student is to validate whether supercomplex formation and sarcolipin are really responsible or behind this cold and obesity resistance in AJ mice. So we have several cool ideas how to test this, and well, we can discuss them in more detail if you sign for our tour in the afternoon. The other project is more clinic oriented. It's based on our collaboration with ICAM. In ICAM, they do a lot of open heart surgeries, and while doing this, they are collecting some sample for us. So we got some biopsies of myocardium and epicardial adipose tissue, and we also collect pericardial fluid and plasma, peripheral plasma from these patients. And then we can analyze these samples using multi-omics approaches. And this actually already resulted in this output by my colleague. But now we want to focus on extracellular vesicles excreted from adipose tissue, especially this epicardial adipose tissue, which is in intimate touch with myocardium, and thus can really affect what's happening in the heart, because it was shown that extracellular vesicles secreted from adipose tissue can contain even mitochondria, potentially dysfunctional mitochondria if the adipose tissue is obese, inflamed, and so on. And these extracellular vesicles can be taken up to the heart. And this was shown for normal peripheral adipose tissue, but now we have these unique samples from epicardial adipose tissue and pericardial fluid, so really from the adipose tissue, which is just next to the heart. So again, the plan is to isolate extracellular vesicles from these samples from ICAM and to do several analyses, especially this proteomic analysis. So that's everything for now. I thank you for your attention, and maybe I'll see some of you in the afternoon.
Thank you very much, Petr. And next presentations will be from the Laboratory of Bioenergetics. Tomáš Mráček and Petr Pecina will present 4 topics for all levels of studies from bachelor to PhD.
Yeah, okay. So yeah, again, thank you for the introduction. And I'm from the Laboratory of Bioenergetics here at the Institute. And we are like a bit larger group with 4 PIs, and actually every one of us is now potentially searching for the PhD student. Also, it's just 2 of us who will be speaking, but if you want to meet anyone like in the afternoon, we are available in the laboratory to discuss details. And as you can see, we do have a group of PhD students and master students, but since Christina just defended and Alexandra and Zuzka are about to finish by the end of this academic year, it means that we are really potentially hiring a bit more PhD students than usual because there will be places available. Like we are quite international group at the level of PhD students, with students from Spain, Mexico, Czech Republic, Serbia. So we are really open to everyone. And yeah, obviously our laboratory studies this particular pathway, which, as you can see, is central to the energy metabolism of the cell, which is obviously a joke. But if you think about the mitochondria, like obviously you have to think about the metabolism in general, because mitochondria as organs are definitely classically providing the ATP for the organism, and that's probably their major role and why they evolved from the alpha proteobacteria to be the organelles in our body. But since they do have like specific environment from the redox perspective and from the perspective of pH and so on, many metabolic pathways in the end during the evolution placed like some of their reaction into the mitochondria because it was simply easier to make this particular reaction inside the mitochondria. But that means that if something goes wrong with the mitochondria, obviously you screw up a lot of other stuff. And this is why the mitochondria are so important, not that in many metabolic pathways like they happen there, they may have just one reaction going on there, but if you screw up the respiration, mitochondrial respiration, which generates the potential and keeps everything in check in the mitochondria, you screw up a lot of other stuff. And it's also like what we do study in the mitochondrial metabolism in our laboratory, it's primarily the respiratory chain. So that part of mitochondria which builds up the membrane potential across the inner membrane and then utilizes it for the ATP provision. We come to this topic mostly from the area of inborn errors of metabolism and mitochondrial disorders, which are the disorders where you have mutation in the patient in some of the subunits of those complexes or in other proteins which help to assemble them, which help to deliver the particular cofactors to them. And obviously all those mutations then manifest as a disease. Initially, we were involved in characterization and validation of the disease-causing genes, which are still some projects in the laboratory, but now we are looking more into the details, what this causes further in the metabolism and whether there are not some possibilities how to interfere and mitigate the phenotype and potentially deliver therapy to the patients. So that's where we are. And we have like 2 enzyme complexes which we love most, and it's ATP synthase and the cytochrome C oxidase. And I will start with the ATP synthase, and then Petr will talk a bit more about the cytochrome C oxidase. And so, yeah, the ATP synthase, as all the others, is multi-subunit enzyme which is embedded in the mitochondrial membrane. And it has subunits which come from mostly the nuclear genome, but there are also 2 subunits coming from the mitochondrial genome, which means that even if you screw up something in the mitochondrial DNA maintenance, mitochondrial translation, obviously it translates into the dysfunctional ATP synthase. And that means dysfunction energy metabolism. Again, like purely the defects of the ATP synthase are less frequent than the defects of the respiratory chain, but still they are a relatively common cause of the mitochondrial disorder and quite often very severe one. And typical presentation are cardiomyopathies or neuropathies. Now I want to talk just about one subunit and one protein is this small guy in a red in the whole structure of the ATP synthase is the genes USMG5 or the DAPID subunit, or it has several other names, but who cares? As you can see, it's just single membrane span subunit associated with the ATP synthase in the membrane region. And again, why we study it? Because there is a patient with the mutation. And if you check the patient phenotype, and yeah, the patient was not described by us, it's paper by other people. There is the Lee syndrome. So neurodegeneration, basically your brain is eaten alive and you are losing a lot of the tissue there, and this is the very severe phenotype. The other phenotype is that there is structural difference in the mitochondria, even in the muscle. And I should say this is phenocopyed in the animal models. And if you check what happens to the enzyme, nothing that much. Like you only can see that there is less of those dimers of the enzyme. Monomers are there, dimers not that much. So there is something going on with the structure and with the interaction of those enzymes between each other. This is patient, this is the animal, so perfect match. But not in the whole body phenotype, because as I said, the patient is severe neurodegeneration and dies, while the animals are generally smaller, but also leaner, resistant to high-fat diets, showing better insulin sensitivity. If you check the plasma metabolites, they look just healthier. So they are just happier, and not having DAPID, if you are red, seems to be like perfect idea contrary to the patient. Even like if you check the cellular senescence, so there is less senescent phenotype in the fibroblasts isolated from those animals with the knockout of DAPID. So really, like you'll probably live even longer than the control. So you can either just say that, well, animals are not humans and you can't compare them, but we believe this is perfect starting point for a project, because first of all, like maybe, and we want to test, and we do have funding now, whether simply modulating the mitochondrial activity and slightly decreasing the mitochondrial activity cannot be protective from the point of view of the metabolic syndrome. So basically challenging the idea that you need the mitochondrial function and defining that slightly downregulating it might be beneficial. So that's one possibility. And obviously, there is the other possibility, basically claiming that the animals live in the golden cage, and we only need to challenge them by some physical activity or other environmental stressors to present with the classical phenotype which we observe in the patients. So, like the project should follow 2 ways. First is trying to prove that mitochondrial dysfunction is potentially good for the metabolic syndrome. The other is like doing the contrary and showing that the physical activity, which the patients and all the humans have to withstand, actually prevents this as a potential treatment, because maybe it will lead to the severe disease phenotype even in the animals. So that's one project. The second project concerns this von Willebrand 8 domain-containing protein, VWA8. It's a project by Lukáš Alan. And again, there is a patient. There is a patient with the mutation in this protein. It behaves as a classical mitochondrial disease patient. So there is the lactic acidosis and developmental delay. So worth studying and worth preparing the animal model. And again, the animal model is slightly different to what we see in the patient, because if you check the animal model, there is some signature of fatty acid oxidation dysfunction. Not that strong one, but it looks like there might be something about the fatty acids, which is not case for the patient. And also, like when Peter Zohar before me spoke about the adipose tissue and the white and brown adipose tissue, with the brown adipose tissue having more mitochondria, those adipose tissues can partially switch between the phenotypes. And actually, if you have increased mitochondriogenesis and increase in the content of the UCP1 protein in the white adipose tissue, you may end up with something called the beige adipocyte, which contains mitochondria, has the uncoupling protein, so it can provide heat and also dissipate the energy. And it looks like exactly this is happening here in the knockout animals for the VW8, because we do see more mitochondria in the knockouts, and we see accumulation of the mitochondria in the adipose tissue, which has small vacuoles and is of this beige origin. So potentially, again, like we may have the proof that like modulating activity of this protein can induce the energy dissipation and might be again beneficial in terms of the obesity and metabolic syndrome. So this is about what you should study in case of this project. And now it's case for the PET or task for the Petr Pecina to continue with the cytochrome C oxidase.
So good morning. So cytochrome C oxidase is another beloved enzyme in the lab, and it's great to demonstrate the kinds of questions we research and ask at our department. So cytochrome C oxidase is a obviously very old enzyme. You certainly have heard about it in biochemistry lectures. So this is the last steps of oxidation of nutrients in our bodies. So this enzyme catalyzes the transfer of electrons from cytochrome C to oxygen. And basically this reaction is remarkably conserved. So it occurs the same way in prokaryotes as well in eukaryotes. So this is not something that makes us different from bacteria. What perhaps makes us different is this like cloud of extra polypeptides that are only present in eukaryotes. And they certainly contribute to the fact that the life on Earth didn't get stuck at the unicellular stage, but we could develop into multicellular animals with different tissues, with different physiology. So you can view cytochrome C oxidase as a set of Lego bricks, which you can put together in a different way to obtain a differentially functioning enzyme. So as I mentioned earlier, there's this catalytic core that always stays the same, but the extra subunits that are transferred to the mitochondria from cytoplasm, they are encoded in the nuclear genome, can exist in different variants, and they can give rise to enzyme that works not the same in different tissues. Also, these polypeptides can ensure that activity of cytochrome C oxidase can be modulated by signaling pathways or according to the tissue where they are expressed or the stage of development when they are expressed. To give you an example from the past, so we've been studying one of the major regulatory subunits of cytochrome C oxidase, subunit 4, COX-4, and we found out that if you express different variants, there are 2 variants of this enzyme, you end up with a cytochrome C oxidase that has huge difference in oxygen affinity. So normally you want to have your cytochrome C oxidase to bind oxygen as effectively to allow oxidation of nutrients in your mitochondria, but there are certain specialized tissues where you can modify cytochrome C oxidase to an oxygen sensor, in other words, to an enzyme which binds oxygen with less affinity, not that effectively, and can kind of sense the level of the oxygen in the body. And that happens in lung and in carotid bodies, where there are specialized cell types which modulate physiological response to hypoxia. So in lung, you have special muscle cells that encircle the veins, which ensure blood oxygenation. And by a process called pulmonary vasoconstriction, you can redirect the blood flow throughout lungs to areas where there is the most oxygen and the oxygenation occurs most efficiently. And cytochrome C oxidase in these muscle cells works as a sensor. And if there's not enough oxygen, the respiratory chain stops and you generate certain metabolic signals that trigger the constriction of these muscle cells and redirection of the blood flow. Similarly, in glomus cells, which are cell type in the carotid bodies, which are the essential, which is the essential organ to modulate the systemic response to hypoxia. Also, a cytochrome C oxidase with modified subunit composition is expressed, and also under hypoxic conditions, this enzyme stops and generates metabolic signal that trigger the neural response and make you hyperventilate and supply your body with more oxygen. Currently, we are exploring another nuclear encoded subunit of cytochrome C oxidase, COX-6B, which also exists in two variants. One is canonical and basically ubiquitous and ensures normal activity of the enzyme. But male reproductive tissues, testes, and sperm cells express a specialized form of this subunit that currently we explore the hypothesis that this special type can be modulated by post-translational modification and work as an on and off switch in sperm cells to modulate the activity of these cells before and after capacitation to ensure that these cells efficiently fertilize female eggs. Also, this special type of COX-6B subunit was found out to be expressed in certain carcinoma types. So we think that this feature, that this subunit can be modified by post-translational modification, can be used by cancer cells to facilitate their growth. So another question we study in the lab is how these molecular machines are put together, how they assemble. So if you look at the respiratory chain, so like cytochrome C oxidase, also the other complexes of OXPHOS, complex 1, 2, 3, and ATP synthase are composed of numerous subunits. So to organize the functional OXPHOS in human mitochondria, you need 13 polypeptides encoded in mitochondria, 70 subunits which are encoded in nuclear genome. And in addition to that, you need hundreds of other proteins that make sure that all this assembles properly. And in many of these so-called assembly factors, there are genes that are mutated in human disease. And one of them is SIRF1, which is a specific assembly factor for cytochrome C oxidase. And like the DAPIT gene, which Tomáš was talking about, also dysfunction or mutations in SIRF1 are causing Lee syndrome, that severe neurodegeneration at the brainstem. So this is a topic that's perhaps open for an enthusiast biochemist who would like to study what is the precise role of SIRF1 in the process of putting together cytochrome C oxidase. We think that SIRF1 really controls how and which type of specific heme cofactors are delivered into the enzyme so it can function properly and be stable. Also, we are looking for molecular identity of several other assembly factors which are known in more simple organisms like yeast, but haven't been found out in mammals like CoA2. So we have several candidates, and really we would like someone to come and use models which some of them we already prepared and get to work. So in addition to study how these modified or diseased enzymes work, there's another huge topic which is supervised by Alena Picinova in our lab, and that concerns how cells adapt to dysfunction of oxidative phosphorylation. So all of you probably spent hours and hours memorizing the normal flow of metabolism, the normal flow of TCA cycle, how it goes intermediate by intermediate so that mitochondria produce enough ATP. But if you disrupt anything in OXPHOS, you can forget all of this because the whole metabolism can rearrange remarkably. So suddenly parts of your TCA cycle run backwards and everything has to change so that you're able to produce enough ATP elsewhere than in mitochondria, and also get rid of some perhaps toxic metabolites. So it's important to understand these processes, perhaps to find out new markers for human diseases, or to be able to identify toxic metabolites that we need to get rid of to treat diseases, or to understand which pathways we should perhaps enhance to help patients which have some defect in oxidative phosphorylation. So at the end, I would like to show that if you would like to join our lab, you can be sure that you can get your hands wet with some typical or classical biochemical procedures like protein electrophoresis. You can monitor mitochondrial function by classical Clark electrode measuring oxygen consumption, but you can also use modern methods like proteomics to analyze the levels of the whole proteome in cells and tissues from animals or tissue cultures, or you can analyze metabolites by metabolomic unit, which is available at the Institute. So, you know, if you are enthusiast biochemist or if you're more a data geek, we can make everyone happy in the lab. And I would like to invite you to come and check out the vibes of everyday work in our laboratory in the afternoon. Thank you.
Thank you, Peter. And I would like to welcome the next speaker, Ondřej Kuda, who is the head of the Laboratory of Metabolism of Bioactive Lipids, and he will propose his project.
Well, good morning. My name is Ondřej Kuda, and I'm really the head of the metabolism of bioactive lipids. We are interested in anything related to lipid metabolism. We work in the fields of physiology, biochemistry, molecular biology, and bioinformatics. And there are 6 of us, and I'm the oldest one there. I would like to talk about 2 projects in which students like you could be involved. The first one is a human breast milk project. You can read this annotation online. So I would rather focus on the hypothesis behind this. There are 3 sources of human milk lipids. The mother eats diet. There is synthesis and degradation of lipids in the adipose tissue and also in the liver. And there is synthesis of lipids within the mammary gland. And these 3 processes create human breast milk. We are interested in the composition of the breast milk, what is inside, what type of lipid, how many of these lipids, what is the concentration of these lipids, and whether these lipids are good, for example, for infants who were born preterm or interim. So what is the best combination? What is the formulation of the milk, of the human milk, which can help those preterm newborns? And to decipher this, which means this is the biochemical pathway behind the production of milk. We are focused on the enzymes, for example, fatty acid synthase and genes that encode these enzymes. And we would like to decipher this whole mechanism. To do this, we use biochemistry mainly here. We use tracer, deuterium oxide, which is stable isotope. And using this tracer, we can really get fluxes through these metabolic pathways. And then at the end, we will know what is the metabolic source of individual lipid species in the milk. And then we can, for example, change the diet of the mother who delivered preterm to help the baby to survive the next period of life. You see that this project is quite complex. However, we can divide it into sections, A, B, C. So the level A is good for bachelor's degree, A plus C is good for master's degree, and all three together, they make sense as a PhD level project. If you are interested in human milk and lipids in this milk, recently we published a paper describing new lipids in human breast milk, which are important for those preterm infants. So this was the first topic. The second one is focused on the metabolomics. Again, this annotation is online, so I will skip further. Recently, we published something like this. We call it Metabo Atlas. It's basically an atlas of mouse tissues, all the metabolites within 13 different tissues. And we were interested in the way how these tissues communicate each other. What metabolites are in circulation, what are tissue-specific metabolites, and what metabolites work as the signals. So based on this, we created this pipeline, and we are now using this pipeline, and it's the base for the second project in metabolomics. Okay, this is the project parts. And in this project, we add something more. We add the dimensions. In this section A, we add 3 different dimensions. The subcell dimension, we are interested in how the organelles communicate, what metabolites they exchange and why. Then intercellular level, how different cell types communicate, and then how the organs communicate using metabolites. And currently we measure at about 2,500 metabolites. When we are done with this, we will add another dimension, which means time, because it is important to know when these reactions happen. Then another dimension, which is the tracer, which means flux analysis. We need to know what is the mass unit, how many metabolites are produced or degraded through these pathways, interorgan or intercell or interorganelle. And at the end, we have a model. In this case, the model is either obesity or diabetes, and we will apply this on the model. And this is a collaboration with ICAM on the obesity and heart failure. As before, it can be divided into sections. Something is good for bachelor's degree, something is good for PhD degree, and it is very complex project. We are especially looking for students interested in physiology and animal surgery, because we do a lot of these things, infusion, catheters, operations. And then students interested in bioinformatics, because we generate a lot of omics data, proteomics, metabolomics, lipidomics, transcriptomics, and then we have to handle these data sets. So preferentially someone interested in physiology and or bioinformatics. If you think this is a topic for you, you can contact me, or you can sign up for the PhD program at IFIS or visit us in the afternoon. Thank you.
Thank you, Andrea. And the next speaker will be Michaela Tencerova from the Laboratory of Molecular Physiology of Bone.
Thank you, Olga, for introduction. Thanks, everyone, for keeping time. So I hope also with introduction of this topic that we are working on in our lab. I'm gonna stay also in the track, explaining why we are interested also in bone physiology, why it is important. Maybe also you never heard about this depot before and why it is important to study something that we don't even see, that is in our bones, and it's developing over the time with aging, but also with other pathological function. And thanks to my colleagues before, you heard about different type of adipose tissue and how they are important in regulation of the full body metabolism, and that to have a functional fat depot, it's very important. But of course, if it's too much of this depot in which location, it's also very important to understand in which context we are actually studying this depot. And one of them is also this adipose tissue in bone marrow, which so far, it was so much overlooked in biopsies of a lot of pathologists, because they actually call these cells like white ghosts. Because they didn't know at the time, to explain what it means. But it seems this higher attention to this depot, getting more scientists interested, to studying it. And as you can see, comparing to previous other depots, as you heard about white or brown adipose tissue, there is not much known. So that's why we are also interested in this depot, and where it is coming from is the compartment in the bone marrow where you have these mesenchymal stem cells, bone marrow mesenchymal stem cells, which are multipotent stem cells, and they can differentiate to different cell types like osteoblasts or adipocytes. And it's important that these processes are in good regulation, because of course, if your bone marrow, it's not making a lot of bone cells, so you're getting more fragile bones, and also losing bones. It's not good for our healthy aging in future, if you want to look at the bigger perspective. So in osteoporosis or actually other metabolic disturbances like diabetes, obesity, starvation, or anorexia, actually this depot is increased. So we also found, and other colleagues, that the obesity causes also the higher accumulation of the fat in the bones, and it's contributing to regulation of the full body metabolism. But in terms of context of the bone health, there is a paradox in terms of the bone mass, and how with obesity this process is regulated, because with the topics with the different ways how you can measure bone mineral density, in obesity we can see higher bone mass but the higher fragility. So what it means that there is something going on in the bone marrow, and our studies show that actually these bone marrow cells, which are actually the source of these adipocytes in the bone marrow, have affected their differentiation capacity. They actually have increased adipogenic potential, which is associated with the accumulation of ROS and senescence. So this is also one of the features that we found in previous studies that we would like to follow up on these findings, how we can actually regulate this bone marrow adiposity to improve our bones and also contribute to better physiology in our body. So in our lab, we are actually interested in different animal models, but also with collaboration with our colleagues from clinic. We try to also bring this research in translational impact, that with different interventional studies using different anti-diabetic drugs or modulating your lifestyle, through diet or actually exercise, if you can modulate this depot. But of course, how to measure all these changes in the bones. It's easier in mice. It's not so much easy actually in a human, because to see in the bones, it's not as they are so dense, easy. So the other topic that we are actually working on is also using a different animal models. They have different, a response to high-fat diet. They can be more prone or more resistant to obesity, but they have also different, accumulation of this bone marrow adiposity. That could also give us much more, answers to hypotheses how these processes could be regulated and connected to the bone fragility. So this is also one of the projects that we are working on in the lab, and then we have also like new candidates that we want to test in this context, and using different specific cell-specific knockout animals, like to answer these questions. So, but I mentioned also different type of approach, how to actually to study this bone and fat metabolism. So, as I mentioned, in animal models, we are using, of course, different approaches to study their metabolic activity, but also the bone structure. That it's through using different machine, like imaging machines, because it's very important to see how the structure of the bones is changed, and also important for their strength. So it's using DEXA scan or micro CT, looking at the the different modulation of this dynamic in the bone remodeling. And also, as I mentioned, it's important to see inside of these bones. So we have also collaboration with our colleagues abroad in Leuven University in Belgium, where we using different contrast agents to look at the structure of these bones. There you can see nicely actually these lipid droplets inside of this bone marrow, or based on different type of these contrast agents, you can actually visualize even the vessels. These are like more robust analyses through different imaging and software system. But if someone is also interested in this kind of approach, it's a really nice project, because this also tell us about what we can't see, like, by from the first sight, but it has actually impact for future for our mechanical, loading that everyone actually goes, like, with, dealing with losing actually the high bone quality. And the other approach that we are using is also look at the resistance of these bones to break. So there is actually this testing using 3-point bending, where you have actually bones in mice, or like, they are already actually isolated from mice. And then we are testing actually their resistance to get break. Like, that's one of the quality measurement. And as I mentioned, we are also interested in the quality of these bone marrow stem cells. So we are using, like, the source even from these bone aspirates from patients, or like, from mice, where we are looking actually their different, metabolic activity, or, differentiation potential, and using also different approaches of omics RNA sequencing. And actually also today, if you would be interested to look at, like, how it looks like in our lab. So today we are getting actually samples from, clinic, that we are going to isolate these bone marrow cells from patients involved in our study. And this is our team. It's international team, and we have also a lot of collaboration abroad. So, like, if you join our lab, so we will have opportunity to go for internship to other labs to learn new techniques, and also, like, to visit different conferences that would bring also this networking. Thank you.
Thank you, Misha. And last speaker and last two topics for PhD study will be presented by Veronika Obšilova. She is head of the Laboratory of Structural Biology and of Signaling Proteins. And she is one of the members of our institute whose laboratory is located in BioSef, not here in Krč, but in BioSef. And I will tell you more about how to visit her laboratory after her presentation.
So hello, everybody. I hope you are not too tired since my presentation came the last. So as Olga already introduced me, we are located in this red building in BioSef in Vestec, and it is actually a good place to do structural biology, as we are structural biologists, and there is a lot of core facilities in terms of crystallography, mass spectrometry, and so on. Also, I'd like to stress out that we are not working with the animals. We are working with proteins. So our research focus is that we study protein-protein interactions and their roles in selected signaling pathways like apoptotic, stress, and calcium-triggered pathways. Our current projects include regulation of ubiquitin ligase, NET42, several transcription factors, protein kinases, ASK1, CA, MKK1 and 2. And today we are looking for 2 PhD students and the topics will be these red ones, NET42 and ASK1. So in our lab, we use integrative structural biology approach. We combine the methods of molecular biology, biochemistry, so cloning, mutagenesis, protein purification is our everyday job. Then we can measure activity of the protein. We use fluorescence a lot, analytical ultracentrifugation. And of course, we are most interested to get the structure. So for that, we use protein crystallography, cryo-EM, and NMR. And of course, we use also a lot of proteins are hard to crystallize, so we have to use alternative approaches, which are hydrogen deuterium exchange coupled to mass spectrometry and combined with chemical cross-linking, SACs, and modeling. And with all these methods, we can get information about the oligomeric state of our protein, about binding stoichiometry, about post-translational modification, structure-function relationship, binding interfaces, binding affinity, solvent accessibility, conformational changes, and structure. And together, that is the integrative structural biology approach. So our aim is to get soluble and active protein from E. coli. So in our lab, we are well equipped for cloning, expression, and purification of recombinant proteins. We have 3 levels of these shakers possible to express in different temperature. And advantages of E. coli expression is that they are easy to genetically manipulate, relatively inexpensive, and the expression is rapid, usually overnight. So, so why is it good? What is the preparation of recombinant protein good for? Because if you want to know the structure of some protein, that is the first essential step for to get the protein in solution or in some form. So we purify the proteins. We have 2 of these ACTA FPLCs, and we purify proteins on various columns. This is the typical chromatogram from gel filtration chromatography with the corresponding gels. And here I put several examples of crystal structures which we solved in our lab with the corresponding crystals. This is the example of low resolution structural SACs. First project I am going to advertise concerns ubiquitin ligase NET42. First, I have to start with the ubiquitination. So ubiquitination is the key post-translational modification in cell biology, and so it is a concert of 3 sequentially working enzymes. So we start with E1 enzyme, which is the ubiquitin activating enzyme. Then there comes the ubiquitin conjugating enzyme. And finally, the ubiquitin is then transferred by E3 ligase to the target protein, which then gets degraded. And the specificity of this ubiquitination cascade is dictated by these E3 enzymes, which we are interested in. There are more than 600 of them. We are interested in the family called NET4 E3 ligases. So NET4-2 stands for neural precursor cells expressed developmentally down-regulated protein 4-2. They belong to NET4-like subgroup, which has 9 members, and they are found in vertebrates. They express in lungs, kidney, brain, and heart. And dysregulation of NET4-2 leads to many pathological conditions.
So NET42 interacts or ubiquitinates many ion channels or transporters and signaling pathways. So the dysregulation of NET leads to many diseases like hypertension, kidney diseases, electrolytic imbalance, cardiac anomalies, and so on. That's why that is interesting to really elucidate the exact mechanism of how this ubiquitin ligase work. So here is the domain structure of NET42 ligase. They have the same. They are composed of 3 types of domains. On the N-terminus, there is C2 domain, which binds to the calcium and to the membrane. Then on the C-terminus, there is catalytic Hec domain, and in between, there are WW domains, which are responsible for substrate recognition. And you can see that there are long, the domains are structured, but in between the domains, there are long parts which are really unstructured. So this protein is hard to crystallize. And in these red dots, there are phosphorylation sites, which are recognized by scaffolding protein called 1433, which keeps this ligase in inhibited state. And because this was not the exact mechanism, how 1433 keeps the NET42 in inhibited state is not known. We decided to study this in more detail. So currently we postulated, we built the model of NET42 in the apostate and in the 1433 bound state, just this part missing the C2 domain. And so we found out that 1433 masks these WW domains, and really it affects the structure even of the catalytic Hec domain. And the motivation for further studies should be that NET42 interface can be target for small molecules, and it can have effect for pathological signaling pathways. So what is here waiting for you? So the aims of the NET project is to elucidate regulatory mechanism of other NET4 ligases like SMURF1, ITCH, and others, and understand the role of their binding partners in the regulation of their activity. The second project concerns ASK1 kinase. So what is protein kinase? It catalyzes the covalent addition of phosphate group to the target protein. In general, we recognize 2 types of kinases, either serine-threonine or tyrosine kinases. That phosphorylated residue is serine-threonine or tyrosine, and they play a crucial role in cell signaling. ASK1 stands for apoptosis signal regulating kinase 1. It's member of MAP3K family, and it activates JNK and P38 signaling pathways. ASK1s are activated by various stress stimuli, and they regulate the diverse physiological processes, and they are involved in many diseases, cancer, neurodegeneration, inflammation, diabetes. So here you can see the scheme of ASK1 activation. So upon oxidative stress like DNA damage, calcium influx, bacterial infection, there is accumulation of reactive oxygen species, and ASK1 functions like a sensor, and it gets activated by not really a known mechanism. And the first step in the activation of ASK1 is dissociation of thyrodoxine from it. And then ASK1 activates its downstream kinases. And the final kinases which are activated are P38 and JNK. And the signal is relayed from the outside of the cell to the nucleus. And they finally can activate their transcription factors in the nucleus or mitochondria, and it can lead to apoptosis, differentiation, inflammation, or morphogenesis. So upregulation of ASK1 activity is not wanted because it plays important roles in cardiovascular neurodegenerative diseases and cancer, which you can see over here. So why to study ASK1 regulation? Because its inhibition may provide new strategies for treatment of various pathologies. However, to do this, we need to understand how this kinase is regulated. And the best way, of course, is to understand the mechanism and to elucidate the structure. So here is the model of ASK1 activation. In the resting state, it forms high molecular mass. It's called ASK1 signalosome. It's homooligomerized through its C-terminal CC domains. And in its inactive state, there are two negative regulators of it, which is 1433 protein, I already mentioned, and thyrodoxine. Upon oxidative stress, these two guys part from ASK1, and another protein called TRAF2 or 6 bind to ASK1. And here you can see the active assembly, which is not assembled only by the C-terminus, but also through the N-terminus. There is transactivation of the it is autophosphorylated, and the catalytic domain are activated by transactivation. And actually, these interaction between these two proteins are not known. So we in our lab currently we try to crystallize this complex, this ASK1, and even in the complex, but we started with cryo-EM. And here you can see just this part of ASK1. It's a dimer, and there is asymmetric assembly of central regulatory region. And there are extensive interdomain interactions which are likely responsible for allosteric regulation of kinase activity. And so what is waiting for you? That aims of ASK1 project is to understand how thyrodoxine and 1433 keeps ASK1 in the inhibited state. It's not completely understood. And also to elucidate the role of TRAF binding in ASK1 activation. And for that, we will use the integrative structural biology approach. We also have some international collaboration. Our biggest collaborators are from Innsbruck. We have some small collaborations in Netherlands, in France, and in Germany. Finally, this is our group. You can see that our group is formed mainly by young people. It's actually a fun group which hang out together, not only in the lab. So we are only 3 scientists, including myself. We have 4 PhD students. One of them is Indian, and actually he has to finish this year. And one of them is Serbian. We have 2 technicians and 4 master students. And we are looking for 2 new PhD students. Unfortunately, you cannot visit our lab because we are located 5 kilometers from here. But if you are interested in what I was talking about, just come to me or Google our web pages, and I will tell you more. Thank you for your attention.
Okay, thank you all presenters today. We heard 18 different topics and from 12 different laboratories, and that you can join. And I think now we can close the online session, Tomáš. We thank all that were connected online. The whole morning session was registered and will be available at our webpage until 20th of March. And if you want to listen some topics again, you can do it via this recorded file.